for inviting me to give this talk. There you go. So I'm going to try and convince you not to go to space. Okay, so this whole program started 4 years ago in collaboration with Susie Sanello at the Human Research Program in Houston, who reached out to me, and we started a program to look at accelerated aging and other risks associated with space exploration, including sensory-motor derangements, cardiovascular deconditioning, an increased risk for heart attack that astronauts typically suffer from, sarcopenia, reactivation of a variety of viral entities, and risk of cancer. And we're particularly focused on brain aging, heart aging, and immune decline. We recently published 5 papers in the Nature package that came out in June this year, and we've been discussing at length the accelerated aging hallmarks during space travel, as well as in humans on Earth when you take the cells and subject them to simulated microgravity. And I'm going to explain that. That's all in blood, right? So we envision a way to take human organoids and subject them to microgravity. So we have 3 types of organoids: brain organoids that are derived from iPSCs. Some of these things we make ourselves, some of them we buy. Cardiac organoids that are beating, that are functional. Tonsil organoids are primary from tonsillectomies. And we subject them to 24-hour microgravity simulation in this machine that was invented by NASA engineers to simulate microgravity that keeps the cells in free fall. So weightlessness simulation. And then we do functional assessment as well as bulk sequencing. And we map that back to aging profiles. We do disease enrichment analysis and drug repurposing to identify drugs to reverse or prevent those changes. And as a reference population for the organ aging, we use GTEx, and this was discussed yesterday by Andrea, so I'm going to just skip through. But we've been analyzing and comparing our organoids against this data set. And so what you can see in the plot on the right is a comparison between our human neural organoids and other tissues for which we had enough samples. And it is very obvious to notice that the human organoids resemble human brain the most. And so this is a validation that our organoids are actually recapitulating brain aging, brain tissue. When we subject those organoids to simulated microgravity, we see a whole lot of changes, around 1,600 genes that are downregulated and 700 genes that are upregulated. Many of these genes are associated with aging pathways, including the oxidative stress response. And when you look at more broadly at pathway analysis, the protein, unfolded protein response is largely affected, as well as metabolism and mitochondria. And this is very important for the next part of the presentation, where I'm going to point out the particular relationship between the protein, unfolded protein response and Parkinson's disease. So as far as the hallmarks of aging goes, this plot represents different gene sets that correspond to different hallmarks of aging, the older 2013 version there. And so you can see that things like cellular senescence and genomic instability and the unfolded protein response are all affected. So 3 out of the 9 hallmarks are enriched here when we subject these organoids to simulated microgravity. And just to illustrate the relationship between microgravity and aging, I picked some of the genes that we believe are very important here. So the metallothioneins on top left are small cysteine-rich proteins that bind to heavy metals and participate in the detoxification response. They are also raw scavengers. They increase with age and also increase with microgravity. At the bottom left, we have purine receptors for ATP. They bind to ATP, and they mediate synaptic activity between neurons and between the neurons also and muscle cells. And this gene X that I didn't want to disclose really because we haven't filed any patent yet, so it's not protected, but it's very important because not only participates in muscle contraction, but it's a peptide that's produced by neuronal cells and regulates appetite. And so we have a system that we can use to screen for different compounds, and if we were able to boost expression of this gene, we are onto something very important for obesity and metabolic dysfunction. So to look at more broadly at the changes at the level of the aging clocks, we build a variety of clocks for different sections of the brain. And this is just one example of the cortex. And what you can see in the right-hand side is the age acceleration versus the 1G experiments. And on average, the cells, these organoids, accelerate around 5 to 7 biological age years. When we do enrichment analysis or overrepresentation to look for different diseases, Parkinson's disease is the #1 disease that we're recapitulating here. So Parkinsonism and Parkinson's disease, synucleinopathy more generally. And we validated this by qPCR. And there's a couple of genes that I want to pay closer attention to. So one is Parkin, that you can see is downregulated in microgravity as compared to 1G. And synuclein beta. And so Parkin is a very important gene that was named Parkin after finding this mutation is an autosomal recessive juvenile Parkinsonism. It's a paper that was published in Nature in 1998 by this Japanese group. And after that research, a number of publications have identified novel functions for this gene, including the ubiquitin proteasome pathway that takes care of eliminating misfolded proteins, is also participating in the regulation of mitophagy, so damaged mitochondria. If you don't have an active Parkin, they accumulate, and so that's not a good thing. And they and that's what we see, a downregulation of Parkin. We also see a downregulation of beta-synuclein that is critical. Lower levels of beta-synuclein are associated with accumulation of alpha-synuclein. That is one of the causes of Parkinson's disease. So now we're identifying new biology and targets that we can possibly then find solutions for. So similarly to the story that I just presented on neural organoids, we've been using cardiac organoids. In this case, we also included video microscopy and electrophysiology to measure function. And as you can see at the bottom, our cardiac organoids resemble most the heart, which is a good thing. And then what you see in this plot, it's a little bit busy, is that when we put the cardiac organoids in microgravity, there is a very significant decrease in the rhythmicity. So the beats per minute go down dramatically, as shown here, and the contraction time increases, as shown right here. And this is similar to what happens with aging. Every 10 years of normal aging on the ground, there's about 1.5 beats per minute that decrease in our heart, and also in space. But this observation is only true in the constructs that have endothelial cells in there. When we have the cardiomyocytes alone, that's not true. That doesn't happen. So we already identified the cell that is responsible for those changes. And now, moving on to the transcriptomic age, we built another clock for the heart using GTEx. And as you can see in the right-hand side, there is an acceleration of aging in the organoids treated in microgravity. The diseases that are enriched in this case are dilated cardiomyopathy, which is an aging-related disease. The hallmarks of aging are altered intracellular communication, cell senescence, epigenetic alterations, or deregulated neurotransduction again, protein folding with loss of proteostasis. And here we've done some the convolution analysis to identify which genes in the endothelial cells may be causal of that changes that we observe. And those are listed in the right-hand side of the slide. Moving on to human organoids, these we obtained, as I said before, from tonsillectomies from humans, and they resemble most blood and spleen. Unfortunately, we don't have tonsils in the GTEx, but it's good enough. The pathways that are altered are intracellular communication, cell senescence, deregulated nutrient sensing, and reactive oxygen species response or mitochondrial function. And what do we do with these organoids? We test the function. We vaccinate them. We take the live attenuated flu vaccine, and as you can see at the bottom of the slide, they produce antibodies. They produce specific B cells against flu. And when you take organoids from different ages, from subjects from different ages, the production of antibodies goes down with age. So this resembles immunosenescence, but this is done in a dish. So what happens with microgravity? I'm going to show you first this plot right here. Is a rank plot that compares two conditions. So in this case, we're comparing the stimulated with the flu vaccine in the x-axis and the effect of microgravity in the y-axis. And so the way to read this plot is as follows. If you see warm colors in the upper right corner and warm colors in the lower left, that means that in both conditions, the changes in gene expression go in the same direction. So what you see here is that they actually go in the opposite direction. So genes that are upregulated in the vaccine condition now are downregulated with microgravity. So the vaccine response is attenuated in microgravity. And you can actually see that at the bottom of the slide right here. Genes like STAT1, that is a critical regulator of the antiviral response and anti-vaccine response, is upregulated in our tonsil organoids treated with the vaccine, and is downregulated in microgravity. And that's shown here as well. Okay, I think I went pretty fast. It's a good thing because I have to catch a plane. So a few summary key points here, and then I'm going to wrap up. So simulated microgravity acts as an aging stressor on brain, heart, and immune organoids at the level of gene expression. The organoid function is altered, and we can simulate pathology, Parkinson's disease, dilated cardiomyopathy, and immunosenescence. We identified aging disease druggable targets, and the technology, I didn't show you that, but is suitable for the screening of geroprotective compounds targeting biological aging and diseases. We started a company out of this research, the fever of Silicon Valley, and this is a Buck Institute spinoff. It's called Cosmica Biosciences. That's my email there if you want to contact me for collaborations, pharma, licensing, investments, or so on. I want to thank my team at the Buck Institute. Actually, this is a very old photo, so my team has grown. We're now 12 people, but bottom line, they don't do just science. They surf, they go to parties, they go watch the Giants, and so on. And collaborators outside of my lab, and thank you for your attention.
Thank you so much. Thank you, David. Fantastic talk.
Hi. Amazing work. Very good. Congratulations. Have you tested, so do you know the astronauts, the twin astronauts that went to space, right?
Of course, we have all the data.
Some of the analysis we have done in these samples we have had access to showed that the differences they present were only seen after they arrived back to Earth, right? Not during the space flight, but only after they arrived, right? In your tests, did you extended your test after the microgravity system to see the effects after that, or just during that process?
Right. So the first claim is actually not true. For the N of 1 sample, it's very difficult to tell. In Inspiration 4, JAXA-6, and Axiom, that we have all the access to the data, we see changes during spaceflight, major changes during spaceflight. But your question is spot on, because there is a rejuvenation that occurs naturally in these people upon returning to Earth. Right? So and we have the data and we have the samples multiple time points after they return. And so there are geroprotective mechanisms that occur naturally in these individuals. If you think about it, at the molecular level, you can identify all those. At the more physiological level, if you have hard time points or endpoints like ventricular remodeling, that may take 10 years in recover. But in the case of microgravity, we're just starting to do time series during the microgravity exposure and upon return to 1G. So we don't have the data. But great question. Yeah.
Thanks. Hi. Can you hear me? Sorry. Thanks. Me. Yeah. Sorry. Fantastic talk. Thank you so much. So I'm interested in the heart results that you had. Have you had a chance to compare them to real hearts, transcriptome in real hearts?
That's all the work that we did when we compared against GTEx. Yeah. The GTEx database is about 450 samples from postmortem hearts. Okay. And our cardiac organoids, one, resemble the hearts better than any other tissue. And when you build the clock, so this is the transcriptomic clock for the heart. Yeah. We see age acceleration in microgravity.
Okay, beautiful. Thank you.
Yeah.
David?
Or age deviation, I should say.
First of all, for everybody, if you need to protect the effect that we need to solve aging, then space travel, if you want to go to Mars and stuff, that's another reason, okay? Maybe it will impress some people or not impressed otherwise. Listen, when we look at proteomics with aging, right, a lot of the proteins that we are finding are protective proteins. So my question to you is what are the protective pathways that are there? Because those are the ones that you want to increase and protect, right?
Absolutely. This is a very hot topic in the field, as you know. Response to aging, so resilience, protective, and detrimental effects. And I think the genes and the proteins that are part of that protection, it's not very clear which ones they are. But we know that heat shock proteins are positive, autophagy is good, the stress response. So you see that the metallothioneins, I think they increase with age, right? And you see them here, and there's a whole cluster of them that increase with age. They're actually protective. And we see that also in microgravity. Right, so that's the comparison right here. You see the aging effect and the microgravity effect. They participate in the stress response. They are raw scavenging. So that's one example of that one there. Yes, Rafa.
If you look, one of the things that happens when you go into microgravity is that there's a huge change in the biomechanics properties of the membranes. So have you looked at tissues that they are continuously exposed to huge biomechanical stresses like the heart or the muscle compared to tissues that they are not, like the liver or the fat?
We haven't done that side-by-side comparisons. What we've seen in immune cells is that there is a remodeling of the cytoskeleton, and it's major, with genes like CDC42 being completely dysregulated. And so the acting cytoskeleton is acting pretty weirdly. And I think that's the initial step for everything else that happens in the cells, mechanotransduction.
All right. Thank you so much, David.
Yeah,
we have to move on.
Thank you much.